I'm Tess Ruswick, and I was hired by the Southwest Conservation Corps last spring to do a cottonwood suitability assessment test along the Dolores River. And along the way, we also did some active revegetation. So I worked pretty closely with the Tamarisk Coalition and other members of the DRRP, or the Dolores River Restoration Partnership. So I'm just going to walk you through the project and summarize what we found at the very end. The project was initiated in 2009 with the founding of the DRRP. They wanted to restore the native vegetation along the Dolores River, but also be able to collect data that could later inform other management decisions. And so this project can really be broken up into 3 stages. The first stage was the initial invasive treatment. And then the second stage was the stage that I was involved in, which started in February 2014. And that was where we did site assessment and we got to do some active re-veg as well as some groundwater well monitoring. And the third stage was the rapid monitoring, and that took place later in the year of 2014. And I was not involved in that, but I'm going to talk about the results that they found because they're pretty interesting. So the project took place along the Dolores River. It spanned 175 miles of the 250-mile-long Dolores River, so it was a big chunk of it. And it was a pretty exciting project because it involved, like a lot of these collaborative projects that have been coming out in the last couple of years, it involved both private and public landowners, since rivers just don't accept our boundaries that we put on them. So we got to work with a few different public land managers, 4 different BLMs in particular, Grand Junction, Uncompahgre, Tres Rios, and the Moab. So the first phase was initiated in 2009, and there were 3 different conservation hand crew corps working to manually remove the tamarisk. And you can see in this picture, they all have chainsaws and look like they could definitely take on a few tamarisk. Because it was a study, there were several different treatment options that they went ahead with, 50% removal and 100% removal, because they kind of wanted to see the effort involved and the later effects. And this would allow for the establishment of native vegetation. So just in case the native vegetation didn't establish on its own, we kind of wanted to give it a little push and shove and help it along. So in 2014, in February, we started the second phase of the project to go ahead and do active revegetation. So at first, the project was just going to be a site assessment, so we would go ahead and test the soil and water conditions and see if it was conducive to active re-veg. But then we had enough funding and we had the equipment available to dig the holes. We figured we might as well stick in a couple of trees while we're at it too. So we got to do active revegetation as well. At the same time, the excavator that we had could also be used to install groundwater monitoring wells. So we were able to do all 3 things at the same time. So first we did a site assessment. We did 2 different samplings. We did a shallow whip, which was located really close to the riverbanks. And we would dig 3-foot holes to assess the soil and water, the groundwater, right on the edge of the river. And that was for potential willow plantings. But the majority of our time was done, we did deep sampling. And we were testing for water to see the depth of the groundwater as well as the pH and salinity of the groundwater. And we also tested the soil, the texture, the pH, the salinity, to see if the conditions were conducive. So, just wanted to point out, this is our lovely excavator, and it was manned by Stan Young over here. And it was a great help. Obviously, we couldn't do any of this project without it. So it was a big portion. And right here you can see its arm And then there's the stinger attachment, which was 9 feet and was able to poke both the sampling holes and the holes, obviously, for the trees. And then I'll just walk you through the sampling process. You can see an up close and personal shot of the stinger attachment. Once the stinger was poked in the ground and then pulled out, we could see if there was moisture at the bottom of the well. And when there was, it was a hallelujah moment because it was super exciting. And we got to go and test it. And this, I love testing. Everybody does. So over here, Kelly, my colleague, is holding this super handy tool that was the brainchild of Stan and Sparky. And basically, it's just a pole with a cup at the end, which seems pretty simple, but it's simply genius. And that we could put it down the hole. There's markings all up along the pole, and we could figure out how deep the groundwater was. And then we could use the cup to then pull out the groundwater and scrape out the soil for sampling. And then right there, we had a pH and salinity meter, and we were able to test and see if we could plant. And I know this picture looks like I'm very afraid of the tablet, but this is the first day, and it got better. So we had a tablet to collect data, which was really great that we didn't have to keep track of 200 sheets of Right in the Rain. And then we also had this software program called Map It Fast. And if any of you have questions about Map It Fast, I can answer them later. But basically, it was just a way to streamline and all our data, all the data collection that we were doing, the GPS points, and the photos that we were taking. So if all the conditions were fulfilled, then we were able to plant, and it was great and very exciting. We collected a lot of data, but the three things that we were looking for was if we hit groundwater within 9 feet, because that was the length of the stinger, so that was the limiting factor. And since we were deep pole planting, we were planting poles that didn't have roots, and so we needed to make sure that we reached that groundwater level so that it would stimulate the root growth in the cottonwood trees. We were also looking at salinity. Salinity is really important. The soil and the water can't be too saline or else it kills the tree. So the threshold that we set was under 4 microsiemens, and this data we got from the Scher et al. 2010 study. We also needed to have more than 2 feet of dry soil, and because we couldn't just plant in anaerobic conditions. Of course, this was the metrics that we set. We worked with a lot of land managers and landowners, and it was important to work with them and figure out what they wanted. So sometimes we would plant in more experimental spots where it was a little higher in salt or a little dry. This was for 2 reasons. We wanted to work with the land managers, and also it was experimental, so we wanted to see if we could push those limits a little bit and see what happens from there. So after we tested, we then planted the tree. The trees came from Stan Young's nursery, and the seeds came actually right from the Dolores and Colorado River, so they were locally sourced. The excavator over there, who we nicknamed Cinnamon because of its bright, cheerful color, would go ahead, dig that 9-foot hole, and then we would place the tree in and carefully place the soil around it, tamp it, and then cage it against herbivory and hope for the best. And a stage that I don't have up there because we didn't do it until 6 months later, but I would recommend doing it right after you plant that tree, is to tag it. We had to tag the trees afterwards. So the third thing that we did was we installed groundwater monitoring wells. While we did make sure that there is water in the hole, obviously this is a regulated river, and we were planting really close to the water where the water can be easily influenced by the flow of the river. So we wanted to make sure that we had a way to track these fluctuations. and we would be able to diagnose if it was the water level that was affecting the survivability of that tree. So we installed some groundwater monitoring wells as well. And as you can see, yes, you do need 5 people to install a well. Okay, so at the end of the project, we got to work on 125 miles of the Dolores River. We visited 21 sites. We sampled 231 holes, and then we managed to plant 114 trees and install 7 wells just in a month and a half. Okay, so, oh no, it's missing a picture. That's okay. Um, so in July and September, the rapid monitoring commenced, and this rapid monitoring will continue for the next few years to see how these trees are doing and which ones survive. And this one was conducted by the SCC Strike Force team, who is led by Katie Birch and Kristen Meyer. And they found that 49.1 of the trees survived, so almost 50%. And this number could have been influenced by a couple of things. First, we were really working with the land managers, as I said, landowners. So we didn't always abide by the metrics that we had first set. The other thing is when we planted in a very specific time period, February to March, and that was really important to plant then because we wanted to plant before the trees budded out and would allocate all the resources to the leaves instead of the roots. And we wanted to plant when the base line of the river was at its lowest because the cottonwoods need that source of water. So we wanted to make sure they had that source of water throughout the year. However, there are some unexpected irrigation calls on the river, and we think that the river was unusually high when we planted, so it might have been above that baseline, and thus the river could have then dropped, and the groundwater could have dropped below what the cottonwood had access to. However, there were some interesting things that we found. There was a huge range of survival just within sites. Some sites had 0 trees survive, some sites had all the trees survive. So we should be able to kind of eke out different factors from those sites and see what's really contributing significantly. So I just wanted to compare 2 sites that were pretty similar. Both these sites were located on the same BLM jurisdiction, and both of them had similar sampling sizes, about 10, and both of them were done around a couple days of each other. As you can see, boat ramp, all the dots in red, I don't know if those of you in the back can see it, but those dots in red signify mortality, and green are the cottonwood trees that are still living to this day. And on boat ramp, 9 out of 10 trees died, and then on bridge test, we had 5 out of 11. So around the 50% that was averaged throughout the entire study. So I just wanted to throw up some numbers, some qualitative and quantitative factors that might have attributed to these different success rates. At boat ramp, as you can see, there's only a 10% survival. The bottom, whereas bridge test, there was 46 survival. We found that on average, the bottom soil salinity, which is generally the highest salinity of the 3 levels that we took, the bottom, the middle, and the top, was 2.3 versus the 0.83 of bridge test. The same thing with the water. The groundwater was much more saline at boat ramp of 2.6 than bridge test of 1.84. And then boat ramp was dominated mostly by clays, whereas bridge test was more loamy soil, and clays hold that salt and don't have as much oxygen as loamy soils, so the cottonwoods tend to prefer the loamy soils. But an interesting thing to note is that we had that 4 microsiemens threshold, and all these numbers, while being under 4, um, obviously the higher 2.5 microsiemens seem to have a major effect on the survival. And that can be determined more by further analysis. So we found, at first, going into this study, we thought that there are 2 major factors contributing to the possible survival of these cottonwoods. We looked at presence of groundwater, which at first we thought was just going to be a binary 0, 1, but of course when you get into the real field work, sometimes it doesn't always work out this way, and we found that it was more a quantitative measure of either we got water, or we got a soup, or we got more of a sludge or a stew, one might say, of the soil or of the groundwater. And so we noted all of this down, and we could standardize it easily because it was just myself and Kelly writing down this data. And that could have a huge impact. If we found sludge, it was probably an indicator of the groundwater being right there. So if the groundwater dropped any more, it could have dropped from out of the range of the cottonwood. We looked also at salinity, but other possible factors might have been the soil texture, the time of planting. I mentioned that we planted in between February and March, but in between that time, we were looking at 30 degrees and the soil being really, really frozen at the beginning versus in middle March, we were 60, 70 degrees. Some of the trees started budding out and the ground was very soft. So even within that month, there could have been an extreme variability that impacted the survival. Also, we can look at private and public land, right? On the private land, these land managers, these landowners are within walking distance of their trees. They can observe, they can water these trees a lot more easily than, say, public land managers who are working with so much land, and so that might have a contributing factor. I know private land owners had about a 75% success rate. So groundwater level following planting could have been a potential factor. Like I said, we could have had some of the trees dip below that that groundwater, the groundwater dip below the trees if the baseline was actually lower than we first encountered it. Another thing to consider is the mycorrhizal presence in the soil. They found in recent studies that mycorrhizal community is disrupted by tamarisk. And so we were planting on sites where tamarisk had previously dominated. And we didn't end up inoculating the soil with mycorrhizal community. So that could have contributed to the success rate of some of the cottonwoods. So we don't have data on mycorrhizal, but we do have limited data on the groundwater level since we installed the groundwater monitoring wells. So in conclusion, you can look at one small part of the study and the, I guess, more quantitative measure, 49.1%, and try to figure out if that's a successful number or not. But this organization, Dolores River Restoration Partnership, looks at more than just that. They look at the ecological management, economic and social implications of this study. And just running through some of those, you can see how the project really influenced more than just the binary survival of the trees. So ecologically, we managed to plant 113 trees, and 50% of that survived, and those trees will then be a seed base for future passive revegetation of cottonwoods. For management, we can go through this data and really figure out and determine what factors had the most influence for the survival of the cottonwood trees in general. And this can inform later management restoration projects that are very important. And then economically, this project hired young crews as well as local contractors and local nurseries. And then the social implications, we had youth corps with the hand crews. The restoration crew that I was on was an SCC crew, and then the SCC strike team, strike force as well was involved. So it involved a lot of young, hopeful stewards, who I'm sure all will go on to do great work in the future. So I know myself personally, after this job, I went on to work with the BLM in California, and now I'm applied to grad school, and Mike White, my mentor at SCC, wrote me a great letter of recommendation. So it's just been a great process for me as well. And these are all our numerous partners and funders. So if any of you have any questions, feel free to ask them now or after the talk.